Hello, students, and we are back. And I really enjoy this topic, so hopefully you will like it as much as I do. I find genetics to be quite fun and one of the more interesting things that we get to cover in class. So we will get started with that. So we're on chapter 11 and Mendelian patterns of inheritance. And this, again, like I said, is quite fun. I think genetics is interesting to look at. Part of the reason is that I am one of five children, so I have four siblings, and it's fun to compare my genetics to my siblings' genetics and then compare that to my children. So I have four children and looking at what my parents had. And it's just kind of neat to see how that works and the predictions that can be made. So let's get right into it and get started. So we'll be talking a little bit about Gregor Mendel and why he is important and how he helped with genetics. We'll be talking about his laws of inheritance and patterns of inheritance in human disease, and then just kind of a little bit beyond Mendelian genetics too. So first of all, Gregor Mendel, he was a monk who looked at pea plants, and he played a huge role in the development of genetics and kind of the principles that started it. He started looking at how the parents had blending of their traits and their offspring and was able to start predicting what the offspring would look like. And by running many, many tests with his pea plants, he was able to predict the offspring and also be able to come up with this idea of dominant and recessive alleles. So what he noticed is that the blending concept of inheritance is that each parent has something to contribute, and the combination of those two parents helps to determine what the offspring looks like. And over time, the variations would decrease as individuals became more alike in their traits. Blending was a popular concept during Mendel's time, and Mendel's findings were in contrast with this. He said that particulate theory of inheritance, he kind of suggested that there was something going on, that there were these genes present, something present. He didn't necessarily know they were genes at the time that was allowing everything to get shuffled up, but we could still kind of predict what we were going to see from generation to generation. So again, he was an Austrian monk. He studied science and mathematics at the University of Vienna, and he conducted experiments with the garden pea, and he was very precise and very meticulous in his process. He documented everything, and he formulated these fundamental laws of heredity in the early 1860s. 1860s, that's pretty impressive. I mean, we're talking about really advanced thinking for a really kind of long time ago. He had no knowledge of cells or chromosomes, and he did not have a microscope either. So it's really kind of cool that he was able to come up with these ideas just based on his breeding of these different peas. He was able to figure out quite a few things based on the way that he bred them together. So peas were a really good choice for him because they were easy to cultivate. They had short generations, which is something that's really important when we're working with genetics, is you don't want something that has a really long generation time because it's going to take a while for you to see results. If you have something with a very quick turnover, then you'll be able to receive results a lot quicker. Normally self-pollinating, but can be cross-pollinated. So that's what he did. He chose traits that he wanted to test, and he was able to cross-pollinate them on his own. The pollen was transferred from the male anther of one plant to the female or stigma of the other plant. And this created the opportunity for him to do something called true breeding as well. So let's say you had a white plant and you only wanted white plants. So you just bred with other white plants and you got white plants. But what happened if you breed that white plant with a purple plant? Then what's going to result? And that's what he was able to do. Now here we can see just kind of the general makeup of the pea plant. So here is a purple example. Here's the stamen and then the carpel. And he was able to take a brush and lightly dust it on the female reproductive part after cutting away the stamens so that he was in charge of what kind of pollination was taking place. So he cut away the part that would typically collect the pollen and he would pollinate it himself, and that way he could determine what exactly was being produced. Now, he was able to look at traits that had one trait and also things that had two traits. So, starting with the most simple, looking at one trait, we can call those monohybrid. Mono for one, because one trait is involved. He performed these crosses and formulated the law of segregation based on monohybrid crosses. So when we're looking at Mendel's laws of genetics, there's a couple abbreviations. P for parent. So this is the original parent generation. F stands for filial generation, and if it's an F1, then that's the first generation of offspring. So thinking of my family, my parents would be the parent generation. I would be part of the F1 generation. My children, my own four children, are the second filial generation, so they would be the F2 generation. And so it can keep going. I mean, you can have F3 and F4 and so on, but usually what we look at is parent F1 and F2, and we're able to see the results that we can predict what's going to happen. Again, with a monohybrid cross, we're just looking at one trait and really taking into account that one trait. So this formulated the law of segregation. So here we look at tall versus short. So we have a tall plant, which is dominant, and another thing to keep in mind with this. Dominant is usually indicated by an uppercase letter. Sorry, let me try that again. Dominate is indicated by an uppercase letter. Recessive is indicated by a lowercase letter. So in this case, we're indicating that tall is dominant and short is recessive. If an individual has two big T's, this is called true breeding. If you breed this with another TT, big T, you're always going to get a big plant. If you breed it with a little t, this is true breeding for short. So true breeding means that it has the same allele combination. But what's interesting is if you breed a tall and a short together that are both true breeding, what are you going to get? So here's how this works. Remember we learned in meiosis that with meiosis, only one part of the information can get passed down. In this case, this parent can pass on a big T or a big T. It doesn't have a choice. It can only pass on one of these, but since they're both dominant, we don't have a choice. It can only pass on a big T. And this can only pass on a little t because we have two little t's. It can pass on this one or it can pass on this one. It can't pass both, but it's only going to be able to pass on a little t because that's all it has present. So this passes on a big, dominant. This one passes on a little, recessive. And then you get what's called a heterozygous. Hetero for two different kinds. And in this case, we get a dominant and a recessive allele present. So in the F1 generation, we have a heterozygous individual. These were homozygous because they're the same. Big T, this is homozygous dominant. This one is homozygous recessive because it's two little lowercase ones. Anytime we have a dominant present, typically, the dominant is going to override the recessive. So in this case, we see that the plant is tall. It's a tall plant because that is the more dominant allele. It still has that recessive allele. This explains why, in some generations, a trait seems to skip because the trait is recessive and it's being hidden by that dominant trait. So in the F1 generation, we don't see that recessive trait, that short plant. We only see the tall plant. But if we do something called a Punnett square, now Punnett squares are actually it's like a logic problem, okay? So let's say we had true breeding parent that was tall, a true breeding parent that was short, homozygous recessive, homozygous dominant. We take one allele from each. It can only give a dominant. It can only give a recessive. We get a heterozygous. But what if we bred two of these together, two heterozygous? Well, in this case, it could give a dominant or a recessive. So that's when we put it into a Punnett square. We put a dominant here and a recessive here because this parent can give either a dominant or a recessive. If we had another F1 generation, it could also give a dominant or a recessive. So we put that on this side. One parent on one side, other parent on the other. Egg on this side, sperm on the other. Although in truth, it doesn't really matter if you put the mom or the dad on this side or the mom or the dad on this side, you're going to get the same results. But this case, since it's the egg, the mom's genetic combinations are up here and the dad is here. The dad can give a big T or a little t. The mom can also give a big T or a little t. Now the way a Punnett square works is you just drop it down. Big T and big T. That's one combination. Little t, big T. So we can have this combination. Big T and little t. We can have this combination, or we can have two little t's. So we see that that recessive trait reappears in the second generation. It's missing in this first generation. It was present with the parents, but in this first generation here, we have the recessive trait disappearing because it's overridden by the dominant trait. So then it reappears in the F2 generation because now we have two recessive alleles. But what's interesting is we have a combination of a 3-to-1 ratio. This is called a phenotype. For phenotype, think physical. Physically, this is what it looks like. Physically, we have 3 tall and 1 short. So we have a 3-to-1 ratio. Phenotype, physical appearance. Now we also have something called a genotype. Genotype is the genetic appearance. So in the genetic appearance, we have a 1-to-2-to-1 ratio. Genotype is genetic, phenotype is physical. Phenotype is 3-to-1. Genotype is 1-to-2, because these are the same, to 1. All right, so here's what he found. He did a lot of crossing. And no matter how many times he did the crossing, he got the same results, which is amazing and helped him figure out, okay, something's going on here. He crossed tall and short plants, and he figured out that the dominant was the tall, and he always got a 3-to-1 ratio. Look at these results. No matter how many times he did it, 3-to-1, 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 every single time. And he was able to determine, yes, something is going on, because no matter how many hundreds of crosses I do, I am still getting the same result. That 3-to-1 is coming each time. And he was able to determine that a dominant trait is a tall plant with an inflated pod shape, round seeds that were yellow. They had this axial representation, purple flowers, and a green pod. The recessive traits were short with constricted, wrinkled seeds that were green. They have a terminal flower and white flowers with yellow pods. So these are all your recessive traits. And he did this hundreds of times. And all of that just supported his research that said phenotype is going to have a 3-to-1 ratio. So each individual has a pair of factors or alleles for each trait. And we've already mentioned an allele is kind of an alternative form of a gene. The factor segregates or separates during the gametes in the sperm formation. Each gamete contains only one allele from each pair. Fertilization gives the offspring two factors for each trait, one from each parent. It could be dominant, it could be recessive. The dominant is the capital letter and the recessive is the lowercase letter. And it really honestly doesn't matter which letter you pick when doing these crosses, but it's helpful to pick a letter where the uppercase and the lowercase look different. So think of a big S and a little s. Big S and little s, they look a lot alike, and it would be hard to determine which is uppercase and which is lowercase. But something like an L, the upper and lower is different, so it's easier to determine what is dominant and what is recessive. But the letter actually doesn't matter, as long as one's uppercase and one's lowercase. The dominant is the capital letter, the recessive is the lowercase. And the dominant is often, but not always, but most of the time, going to hide the recessive. Alleles occur in a homologous pair for a particular gene. A locus is a location. Think locus, location. So a specific location on the gene is where these alleles are going to be found. As I mentioned, homozygous means that they're identical. Heterozygous means that they are different. So here we can see these are the same alleles at a location, a locus, on a gene. So here's similar alleles. This is an allele for S. This one's an allele for S, T, and so on. So then when we duplicate, so we have this is the exact same as this, and same over here, because replication has taken place. And we've got our sister chromatids. They've still got similar genes. Now, this is a homologous pair like we saw in meiosis. One homologous chromosome and its sister homologous chromosome. Genotype and phenotype, which I already mentioned earlier. A genotype is the genetic makeup, like big T, little t. It is the genetic combination, whereas phenotype refers to physical. PH for phenotype, PH for physical. Gene. Genotype is genetic. So phenotype refers to the physical appearance. Dominant alleles often will hide the recessive alleles, and the dominant and recessive alleles represent the DNA sequence that codes for proteins. So protein synthesis is the creation of a protein, and that is a process that DNA is used for. DNA breaks apart, and through a process called transcription and translation, a protein is made. The recessive allele represents a loss of function, so it kind of hides, and really a lot of times the only way the recessive allele can be present is if there are two recessive alleles that kind of support each other. So during meiosis 1, the homologous chromosomes separate, and the process of meiosis explains Mendel's law of segregation and why only one allele for each trait is in a gamete. So that meiosis process we saw with the law of independent assortment can be explained by the processes that Mendel saw. Now, let's talk about a dihybrid cross. A dihybrid cross is a little more complicated. It's looking for two traits at the same time, di for two. Dihybrid crosses that Mendel performed actually helped him determine the law of independent assortment. And this means that the pair of factors of one trait segregate independently of the factors for the other trait, and all possible combinations of factors can occur in the gamete. Sounds confusing, but let's continue and look at some examples, and it'll help it make sense. Just like with the others, we're going to start with true breeding. True breeding means homozygous dominant and homozygous recessive. If you have a homozygous true breeding dominant and a homozygous true breeding recessive, then that means they're going to have dominant characteristics. So if we look at the parent generation and then the F1 and then again the F2 generation is where we're going to see the most results. So again, a little bit confusing, but let's work through this. Okay, parent generation, true breeding for tall and having trouble for short pod. Okay, so tall plant and green pod versus yellow pod. Those are the traits we're testing for, whether the pod is green, whether the pod is yellow, and whether it is tall or short. So two traits we're looking at, tall and short, green or yellow. In this case, we have a true breeding tall green plant. This case, we have a short yellow plant that is true breeding, homozygous recessive, homozygous dominant. This one can give only a combination of a big T or a big, is that a G? I think it's a G. Having trouble seeing my screen. A big T or a big G. And so then it's making that combination over here. This one can only give a little t or a little g. And so our result is big T, big G, little g. So heterozygous individual. If we take the heterozygous and put it into a Punnett square, it gets a little more complicated because this big T can match with the big G or the little g. This little t can match with the big G or the little g. So we have this possible combination from one parent and this possible combination from another parent. Again, because we're looking at two traits, and it can give big T, big G, or big T, little g. Little t, big G, little t, little g. Those are the combinations that each parent can do. So you put that into a Punnett square that's a little bit bigger than the monohybrid cross, and you compare that. And you're going to get, you bring this one down and you bring this one across, and you're going to get a homozygous dominant. bring this one down, bring this one across, bring this one down, bring this one across, and so on. So you'll see here that we have a genotype and phenotype that's a little different. What we end up with is a combination of 9 to 3 to 3 to 1. And that is the typical dihybrid cross representation, 9 to 3 to 3 to 1 ratio. 9 for dominant, to 3 for this combination, tall and yellow, to 3 for short green, and then 1 for that one recessive trait. 9 to 3 to 3 to 1. That's what we're going to see. And here we kind of see how that process works with independent assortment during meiosis. We've got the homologous chromosomes. They're going to line up randomly. They get separated out randomly. And then we have these possible combinations with the resulting parent, or with the resulting cells that we got from the original parent. Now, the Punnett square is a table listing all possible genotypes resulting from a cross. You can do more complicated trihybrid, or looking at 3-trait Punnett squares, but those get really big and confusing. A lot of times, especially in a basic biology course, we're just looking at a typical dihybrid or monohybrid cross. But all the possible combinations of the sperm and all possible combinations of the egg, and that will tell us all the possible combinations of the zygote, looking at both genotype and phenotype. So if we're looking at a Punnett square, the Punnett square in the next slide is going to show how this works. It allows for you to easily determine the genotype and the phenotype. So going just to this, we've got blonde hair versus darker hair. And we know that this parent is a heterozygous and this parent is a heterozygous. So we can compare the pigmentation here, and we can see that in this case, we could get a recessive allele, and the recessive allele could give us no pigmentation or an albino. So it's possible to compare human traits like this also. And when recessive alleles become apparent in a population, those can be hidden because they were both recessive and hidden by the dominant in the parent population. Individuals with recessive phenotypes always have the homozygous recessive genotype. However, individuals with dominant phenotypes, they could be homozygous or they could be heterozygous. We're not sure which they are. So going back to this one, here's a homozygous dominant. They have 2 dominant traits. This individual is heterozygous and this individual is heterozygous, but they're still showing that dominant trait. However, a recessive allele only shows if there are 2 recessive alleles. A test cross determines the genotype of an individual. So if you don't know if they're heterozygous or homozygous, a way that you can find out is to cross them with a recessive individual. If you cross them with a recessive individual, you can find out based on the offspring what the parents were. Were they homozygous dominant or were they heterozygous? And you can determine that by a test cross with a recessive individual. So here we can see a test cross. We don't know for sure if this is, we know it's a tall plant. It could be heterozygous or it could be homozygous. If it was heterozygous, we have a combination or a possibility of there's some recessive showing up. But if it was homozygous dominant, we're only going to get one thing because this one can only give a big T, this one can only give a big T. All of our plants are going to be tall. Whereas with this one, it can give a tall or a big or little t, a tall or a short plant, and so we can see the appearance of a recessive trait. So there are individuals with both dominant phenotypes is crossed with an individual with both recessive, you're going to get a 1-to-1 to 1-to-1, and that's how the test cross works. If you have a dominant that's crossed with a recessive and it's a homozygous dominant, you're only going to get heterozygous individuals that all show the dominant trait. Now, sometimes diseases can be inherited based on if it is a X or a Y chromosome. Now, In this case, it's a little bit different because think about the X and the Y chromosome. A female has two X's. That means she is only capable of giving an X to her offspring. The male has an X and a Y. So the male can pass on either the X or the Y. It is always the male that determines whether the offspring are male and female. So if a Y chromosome is passed down, then that means that the offspring is going to be male. But if the X chromosome is passed down by the male, then that means the offspring is going to be female. This is also why that if there are genetic diseases that are attached to the X chromosome, it is more often to show up in the male because the male only has one X chromosome present. So colorblindness is an excellent example of this. Colorblindness is a trait that is found on the X chromosome. If a female has colorblindness, she has two X chromosomes. So one of those X's, if they're normal, will kind of compensate for that colorblind trait. But since a male only has one X chromosome, if he's colorblind and that X is affected, then he doesn't have another X chromosome to compensate for that. Males are more often likely to be colorblind because of that. So some genetic disorders are autosomal dominant. Auto meaning that an individual with AA has the disorder, an individual with AA has the disorder, an individual with 2 recessive does not have the disorder. So this is autosomal dominant, meaning that it is present in the dominant trait. Other genetic disorders are autosomal recessive. This one does not have it because it has a dominant allele. This one does not have it because it has a dominant allele. Only those that are recessive do have the disorder. So different genetic disorders can be passed down in different ways. They can be autosomal dominant or autosomal recessive. We can use something called a pedigree chart to help us determine this. A pedigree chart shows the relationship where circle is a female and a square represents a male. A line between the two of them shows a mating, and a line coming down shows any offspring. So these two are offsprings of this parent. We don't know what this parent is, but we do know the mother, because it's circle for female, had a recessive trait. So we know that recessive trait was passed down to these. The father did not have it, so he passed on, and in this case, dominant allele, and 2 females were born. They mated with a male, and you can see how this can be used to predict what the offspring look like. In this case, it looks like 2 cousins actually ended up mating. Probably not a great idea if we're looking at actual people here and not like plants or something else. And so we have some cousins that mated over here, and then the trait reappeared in the population. We can see this happening with some royal families of the past that were often inbreeding because they wanted to keep their royal genes pure. And the inbreeding resulted in traits such as hemophilia to be passed down within a population because they kept passing down those same affected traits. In some cases, they would be a carrier, and the carriers, because they had a dominant allele, they carried the recessive allele, but they weren't exhibiting the disorder because it was hidden by that dominant allele. So if both parents carry one copy of a recessive gene, they are unaffected, but are capable of having children who possibly have copies of the gene. This is why parents could seem perfectly healthy, but they have an offspring that has a genetic disorder, or they have multiple children that are experiencing genetic diseases, because they both are carriers of an unfortunate recessive trait that gets passed on to their offspring, and the offspring receives the recessive trait. Methemoglobinemia, relatively harmless disorder, accumulation of methemoglobin in the blood causes the skin to appear bluish purple. Cystic fibrosis is mucus in the bronchial tubes and pancreatic ducts, particularly thick and viscous, which means like syrupy. So here we can see that coloration we were talking about. The skin is paler, the coloration is not as healthy looking for this methemoglobinium. You can see the coloration is really different in this individual. Cystic fibrosis, this is the cytoplasm of the cell, and we have a defective chloride ion channel, does not allow the chloride ions to pass through like it's supposed to. So the chloride ions are going to build up, and so the lumen of the respiratory tract fills with thick, sticky mucus when it shouldn't, because this channel is closed when it should be open. Autosomal dominant disorders. This is when there is a dominant trait that is passed down. If two parents with a dominant inherited disorder will be affected by one copy of the gene, it is possible for them to have unaffected children if they are heterozygous. They have one dominant and one recessive allele. And an autosomal dominant trait, usually only the recessive children are the ones that are going to not display the genetic condition. Osteogenesis imperfecta is characterized by weakened, brittle bones. Most cases are caused by mutation in genes required for the synthesis of a type of collagen. Huntington disease, a neurological disease that leads to progressive degeneration of the brain cells, and this is a mutated copy of the gene for a protein. Hereditary spherocytosis. Red blood cells become spherical, are fragile, and easily burst. Another pedigree chart to show how this relates. In this case, anything that has a big A or dominant is going to be affected. So we're going to see a lot more affected individuals because that dominant trait pops up a lot more frequently. So this case, we have two parents that are affected, but because they were heterozygous themselves, they were able to produce some offspring that were recessive. The recessive individuals did not have the trait. It's only ones that carry a dominant allele that have the trait. Now, going beyond multiple genes and multiple alleles, sometimes we can look at, like I said, traits that are trihybrid or three genes that are linked together. A really common one that we see where there are multiple alleles present is the ABO blood types. This is also where we can see codominance. Now, codominance means that they are shared equally. So think of it like this. Think of you have a dog that is white and a dog that is brown. If they were mated, you could have a dog with white and brown spots. It is shared equally. The white and the brown traits are codominant. They are shared equally. ABO is a great example of this. If you have a parent that has A blood and a parent that has B blood, it's possible they could have an offspring that has AB blood. The A and the B blood are shared evenly, and so they're, instead of saying, oh, it's A blood, which is dominant to a B recessive, it's not like that. A and B are both equally shared because they are codominant. So oftentimes it is abbreviated like this. I always just thought of it as ABO because it was easier for me to understand. But if you have an A phenotype, you could have big A, or big A and an O. O doesn't have a characteristic that codes for it. So O is just kind of the recessive in this case. Think of O as recessive. If you have B, you could have B and B allele, or you can have B O blood. If you have AB, then that means you have A from one parent and B from another. If you have O, the only way you can get O blood is the two O alleles. Maybe you got it from an AO parent and a BO parent, or maybe you got it from another O parent. I'm a great example of this. I have O blood, which means I had to get the O allele from both of my parents. My mom is also O blood, so that means I know I got one of her O alleles, but my dad was A blood. So that means in order for me to get O blood, my dad had to be this combination. He had to be the A allele plus the O allele. Otherwise, there's no way that I could have gotten an O allele. I got one from my mom, one from my dad. My dad was A blood, so that means he had to be this. Now, looking at my children, I know I have O blood, so I can only pass on the O allele to my children. However, all of my children have A blood because my husband is A. We don't know for sure if my husband is AA or AO, but since all of our children ended up with A blood, I suspect My husband's combination was AA because that was the only allele he had to pass on. He passed on this, I passed on this, which means even though all of our children are A, they all carry that O allele with them. So it's possible that my children could have children of their own someday that have O blood. All right, so heterozygous there's something also called incomplete dominance. With incomplete dominance, this is kind of weird. Remember, with codominance, it's shared equally. So we have that white dog and that brown dog. You combine them together, you get a black and white spotted dog. If you have a white dog and a brown dog, and instead of white and brown spotted, you get kind of this light brown, that's an example of incomplete dominant. The dominant allele is not strong enough to completely hide the recessive allele. So the recessive allele is peeking through just a little bit and not being completely overridden by the dominance. It's not sharing equally, it's just peeking through a little bit. So here's another example, red versus white. If you combine like a red flower versus a white flower that has those phenotypes, you might get a pink intermediate because they are incomplete dominant, which means the dominant is not completely hiding it. So here we can see that we have two heterozygous, and in this case we have two pink. It could end up with a completely dominant and a completely recessive, but our two heterozygotes are a mixture of the two, and that means that's an incomplete. If this was codominance, it would be red and white spotted, or maybe like two petals would be red and two petals would be white or something like that. But because it's a mixture, it's incomplete dominant. All right, we also have something called hypercholesterolemia, and homozygotes for the mutant allele develop fatty deposits in the skin and tendons and may have heart attacks during childhood. You don't really want fatty deposits. That's going to lead to some issues. Heterozygotes may suffer heart attacks during early adulthood, and homozygotes for the normal allele do not have this disorder. Homozygous because this only develops in the heterozygous individuals. Incomplete penetrance means that the dominant allele may not always lead to the dominant phenotype. Many dominant alleles exhibit varying degrees of penetrance. Polydactyl is an example of this. There are extra digits on the hand, the feet, or both. And not all individuals who inherit the dominant polydactyl allele will exhibit the trait. It only happens sometimes. So that means that it doesn't always show in the situation as you might expect. Pleiopatry occurs when a single mutant gene affects two or more distinctly seemingly unrelated traits. This one, pleo means many, and so it can be a little bit confusing. Basically, it's one thing affects many traits. So Marfan syndrome is an example. Marfan syndrome is pleiotropic and results in the following phenotypes: long arms, hands, and legs, a weakened aorta, and poor eyesight. So this one trait results in the effects of many things on the body. So Marfan syndrome affects the connective tissue, and connective tissue can be found in all of these locations. So it's affecting one thing, and this one thing is having many different responses to that. It can have issues with the heart, issues with the eyes, issues with the skeleton and the lungs, because connective tissue is found in all of these different places. Polygenic, poly again meaning many, occurs when a trait is governed by two or more sets of alleles. Each dominant allele has a quantitative effect on the phenotype. These effects are additive, so it kind of builds up and builds up and builds up, results in continuous variation of phenotypes within a population. Human skin color, height, and eye color are examples of these. Our skin has a huge range of variation, and it's not like we fall into this color versus this color versus this color. It's a whole spectrum of colors. Height is the same way. You don't have a person that's 4 foot and 4'5" and 4 foot and 4'6". You've got a wide range of variation, and this is what polygenetic inheritance is that many things go into the hair color, the eye color, or the height, and also some things that are not genetic related. If a person is fed well and healthy, then they're going to grow taller than a person who is malnourished and didn't receive the proper nutrients when they were younger. nature versus nurture sometimes. But with a polygenetic trait, we can see a nice bell-shaped curve. This is called a bell-shaped curve, where the center of the bell-shaped curve is the most common, but we do have outliers on either side of the bell-shaped. There's been just very easy studies done where you sit students based on height in a row, or have them stand in a row, and you can see this bell-shaped curve where most of the individuals are going to fall in this center average portion, but you're going to have those outliers present. So in mammals, the X and Y chromosomes, of course, determine gender. We already talked about that. Females are XX and males are XY. The male is the one that determines male versus female. And so if they give, the male can give an X or Y chromosome, but the female can only give an X chromosome. As I mentioned already previously, there's something called X-linked genes. And in X-linked genes, these are when the X chromosome has some kind of mutation on it. And if it is on the X chromosome, it's going to be more obvious in a male because the male only has one X chromosome present. And in a female, it's going to be less obvious because she could have an affected X chromosome, but the other one is normal and kind of covers it up and compensates for it. So the Y chromosome does not carry a lot of these genes because the Y chromosome is very small and only really codes for that SRY gene we talked about before. So fruit flies are another great genetic organism to study because it's really easy to manipulate their genes, and it's also, they have a very fast genetic turnover. So they have offspring very quickly and they reach maturity very quickly. So we can see variations take place within a short amount of time. So there's been a lot of studies with fruit flies, just like there's been a lot of studies with pea plants. Fruit flies have a similar sex chromosome pattern to humans, and Morgan's experiments with X-linked genes apply directly to humans as well. So here we can see the number of dominant alleles is going to be more common in a population, and the number of recessive alleles will have our outliers, and this is showing skin color. We have few with very light skin color and few with very dark skin color, and a lot here in the middle that are blending of the dark versus light. Fruit flies and the X-linked traits, so you can see kind of how this works. All red eyes, red-eyed, and white-eyed. So if it is a male, the trait can be found on the X chromosome, but the males are more likely to show it because they only have one X chromosome present, and females are more likely to be a carrier in that case. Human-linked X chromosomes, I already mentioned color blindness. Oftentimes males have red-green color blindness because it is on that X allele and they do not have an extra X allele to hide that. Women can have color blindness too, but typically it has to be on both of their X alleles in order for it to show. Menkes syndrome, which is caused by a defective allele on the X chromosome. Muscular dystrophy causes wasting away of the muscles. Andronucleodystrophy is an X-linked recessive, and then hemophilia, which is a clotting factor that makes it so the blood doesn't clot. Here we can see some X-linked recessive. So we have the grandfather here. It shows up in males. It can show up in females if the female has both of the recessive traits present. But most often it's going to be related to the males because it's that X chromosome that's going to show up in males, and females just need one normal chromosome to mask it. All right, well, that's our first step of genetics. We will continue more with genetics in our next chapter. Let me know if you guys have questions. Bye.